the national immunization programs first became properly established and coordinated in the 1960s. By 1900 itself, there were about 2 human virus vaccines against smallpox and rabies, and also 3 bacterial vaccines, that is for typhoid, cholera, and plague. This is in 1900, by 1900 early stages itself. In countries where we had a high vaccine coverage, many of the disease responsible for childhood deaths have essentially disappeared thanks to the vaccines. The number of antigens or the vaccinations, what is used in each country's national program, varies between the countries. The first vaccine which we got used is smallpox, and it was discovered in 1798. And in India, it was, it came to be in use from 1800. WHO estimates that 2 to 3 million lives are saved each year by the current immunization program. The most useful and cost-effective tool to prevent diseases, we all know that, is vaccination. Each vaccine contains either a part of a germ or the whole germ which has been either killed or attenuated. Vaccine not only contains this antigen but also contains adjuvants, stabilizers, and additives. We'll see what the action of each one is. So to prevent contamination, they use preservatives, and the most common one is thimerosal, which is used in flu vaccines. And also to help boost the body's response to a vaccine, they use adjuvants like aluminum salts. They also need to keep the vaccine effective after manufacture. So they use some stabilizers like sugars and gelatin. And also to grow enough of the virus or bacteria while they are making up the vaccine, they use egg protein. And also to kill viruses or inactivate toxins during the manufacture process, they use formaldehyde. And also to avoid contamination by bacteria during the vaccine manufacturing process, they use some antibiotics, and one of them is neomycin. So we know that once vaccine is introduced into the body, so it stimulates the antibody production. The immune system responds to bacteria and virus in a very complex way. It recognizes unique molecules, that is the antigen, from bacteria and viruses, and produces antibodies. Any first encounter with any specific antigen, either in the form of an infection or in the form of a vaccination, some lymphocytes called memory cells develop to confer long-lasting immunity. Actually, this is the basis of any vaccination. So the goal of all vaccine is to elicit an immune response against an antigen, so that when individual is again exposed, a much stronger secondary immune response will result. So we aim these vaccinations against serious and often deadly diseases. So what are the types of vaccines we have? So we have live attenuated vaccine, kill the whole organism, or from the toxin, they make the toxoid vaccines, subunit vaccines, which in the form of purified protein, recombinant protein, polysaccharide, and peptide from the surface of the bacteria or the viruses. And the final one is virus-like particle. So the live attenuated vaccines examples: measles, mumps and rubella, yellow fever, oral polio, BCG, oral typhoid, and Japanese encephalitis. The killed whole organism is whole cell pertussis, that is our DTaP vaccine, and rabies and hepatitis A. Whereas the toxoid vaccines, diphtheria and tetanus, and the subunit vaccines is the most common ones like pneumococcal, typhoid, hepatitis A, hepatitis B, influenza, pertussis. The virus-like particle is the human papilloma virus. So this is a more comprehended thing telling live attenuated. We talked about TB, oral polio, measles, rotavirus, yellow fever, varicella zoster. And we used to have a nasal form of a seasonal flu vaccine, which has been eliminated from the National Immunization Program. That was also live attenuated. Quickly, I would like to talk about polio vaccine. We have 2 types of vaccines for polio. One is oral polio, and the other one is the inactivated injection form. The oral polio is a live vaccine. The oral polio, initially we had trivalent forms, which was active against 3 types of polio viruses, that is type 1, 2, and 3. And the bivalent targets 1 and 3. Now we don't have the trivalent oral polio. We have been moved to the bivalent, which targets only 1 and 3, the wild type 2, even the vaccine when you give, it can cause vaccine-induced paralytic polio. Hence, to reduce the risk of that, we moved from the trivalent to the bivalent from 2000. Another form of vaccine-associated polio is, while you do vaccinate, there can be children or who are babies, they can get the infection as such from the circulating vaccine itself. So IPV advantages, I told you it's an inactivated one, and it can be safely administered to children with the immunodeficient children as well as to premature babies. So that is the advantage of the inactivated injective form of polio. So why we can't do live vaccines in immunocompromised individuals is they will replicate in an uncontrolled manner and they will produce the severe form of the disease in those ones with either immunodeficiencies, whatever type of immunodeficiency, HIV infection, or who is receiving immunosuppressive drugs for various reasons.